Hi everyone, my name is Grace Cannon, and today I'm going to be discussing the therapeutic applications of recombinant DNA and cloning technologies in the breast milk microbiome. When most people think about breast milk, their mind does not immediately jump to a bacteria ridden liquid. They think of the nutritious components of breast milk, the fats and proteins and vitamins, and all the nutrients that are passed to the baby from the mother after the baby is born. Breast milk is necessary for the immune development and metabolism and overall health of an infant. But what many people don't realize is that a lot of the beneficial components of breast milk contribute largely because of the bacterial composition of breast milk. Breast milk is a major hub for bacteria and provides several different bacterial species which benefit an infant and colonize their gut after birth. So I've always been fascinated by this concept. And as I was doing the lab work for this course at the beginning of the term, I became very interested in understanding how the technologies that we're using, the recombinant DNA and cloning technologies that we've used, could be applied to already beneficial breast milk bacteria, and whether engineering these bacteria could make them even better. Turns out a lot of researchers were wondering the exact same thing. Many researchers have started by targeting their understanding of the breast milk microbiome. What exactly is the bacterial concentration? So, as we see in the top right diagram, there are three primary bacterial species that are prevalent in in the breast milk microbiome, the proteobacteria, the actinobacteria, and the Firmicutes bacteria. However, it is worth noting that this image is only one interpretation of the breast milk microbiome. Unfortunately, we don't have a super concrete understanding of breast milk bacterial composition because of various different challenges associated with the DNA extraction from breast milk bacteria. If I can direct your attention to figure 4, the very colorful bar chart, you'll see the work of Douglas and their colleagues back in 2020 when they were studying how different extraction methods of DNA lead to different bacterial concentrations, or at least lead to different determinations of bacterial concentration within the breast milk microbiome. The results of this study determined that even when the same breast milk sample is used, different extraction methods lead to different understandings of concentrations. While the species of bacteria remained consistent, the amounts of bacteria that were recorded in the breast milk changed significantly based on extraction method. This is likely because several of the extraction methods did not account for the presence of gram-positive bacteria in the breast milk, which requires different extraction methods due to the presence of a thick peptidoglycan wall, making it more difficult to lyse. This acts as a very significant limitation in breast milk microbiome research. While there are challenges in determining the exact composition of the breast milk microbiome, there is evidence supporting its benefits. One study of note is shown in figure 5 towards the bottom right of the poster, just above the references section, which highlights the results of a study conducted by Cordy and their colleagues. Their research suggests that bacteria is transferred from the mother to the infant during breastfeeding, which indicates that breast milk serves almost as a transportation vector for bacteria, which help to serve as a natural route for the earliest microbial colonization in the infant gut. This is very important because it indicates that breast milk bacteria can already survive in the human body, which suggests that if genetically engineered, they may be even more effective vehicles for delivering different therapeutic benefits and for therapeutic applications. These understandings are the backbone of the biotechnological applications of the breast milk microbiome. Interestingly, a lot of work has already been done to move forward with this research. In a study conducted by Lin and their colleagues, we see in figure 2, towards the bottom right of the screen, that their goal was to engineer the Bifidobacterium bifidum strain of Bifidobacteria in order to enhance the antioxidant and anti-inflammatory properties of that bacteria. The results of the study indicate that they were successful in doing so, and that using recombinant DNA technology allowed them to improve the anti-inflammatory properties of this bacteria and to express the enzyme catalase, which in turn reduced oxidation within the bacterium. Another amazing example is the Shepler study, where Shepler and their colleagues used shotgun metagenomics, PCR, and SDS page technology to modify Lactobacillus johnsonii to express the PRTB protein. And figure 3 demonstrates that this methodology was successful and that they were able to express this protein. This was crucial because it demonstrated that bacteria that were pulled from breast milk could be genetically modified to express different proteins. In turn, this suggested that genetically engineered bacteria pulled from breast milk could be very useful. Specifically, the L. johnsonii bacteria could be very useful as an oral vaccination method. Another interesting example comes from a study conducted by Young and their colleagues. They aimed to modify the metabolic pathway in L. reuteri by actually deleting a gene, the FOIA gene. This in turn increased folate or folic acid production in the bacterium, which is a very important vitamin for nutrition in the human body because it encourages cell growth. This meant that genetically modifying this bacteria actually did improve its nutritional properties and provide nutritional benefits. While all of these studies do suggest that recombinant DNA technology is very beneficial in enhancing the traits expressed by breast milk bacteria, there are several limitations worth noting in this literature. Firstly, as Douglas highlighted, different extraction methods do yield different results, which means that it's difficult for us to fully understand the exact composition of the breast milk microbiome. Additionally, many of the studies mentioned and many other studies do not use bacteria specifically pulled from breast milk. They use species found within breast milk, but those specific bacteria were not extracted from the breast milk. Additionally, many experiments, and none of the experiments mentioned in this poster, do not use human subjects. Many rely on either literature analysis or mice for testing, meaning that we have not yet moved on to the human clinical trial phase in this research. And possibly the biggest limitation of these studies is that there is very little evidence of long-term effectiveness in colonization. We don't currently have enough evidence to know whether the colonization of these bacteria in the human body would be consistent in the long term or whether it'd be a short-term solution. But despite all of these challenges, there is a lot of evidence that the use of the breast milk microbiome for therapeutic applications is a very promising endeavor. The studies in this review highlight how recombinant DNA could be beneficial in enhancing anti-inflammatory effects or in providing alternative vaccination routes. They could be useful in increasing nutrition or specific vitamin or acid outputs. Due to their natural ability to transfer and colonize into the infant gut microbiome, it also highlights how they could act as sort of vectors or transferring molecules for different drugs or enhanced drug administration. This suggests that the breast milk microbiome could be responsible for creating a sort of living medicine like never before. If I could leave you with just one takeaway, though, it would be this. Although we may think of breast milk as being nothing more than a nutritious meal for an infant, imagine how incredible it would be if we could use biotechnology to make it one of the next greatest medicines.